So welcome back after a wonderful evening. I don't know where you went. I know that the whole crowd who went to the karaoke, so probably they're still sleeping. But anyway, we need to start. Oh, you're back? Okay. So we need to start. So this morning section, we're going to have 3 talks. And the first talk will be given by George Kashanis, right? He's from the Lincoln High, and he's going to tell us about biotechnology, how it is being taught in high school.
Thank you. Thank you very much for the invitation to speak at this incredible conference. I'm absolutely delighted to be here. And I guess I should start by telling you how we got involved. We got involved because of this man sitting right here, Wendell Lim. I got an email in April of 2007 from Dr. Lim, who I'd not heard of, but always answer emails even if you don't know who they're from, because it was loaded with an opportunity. He had invited us to put a team together for the iGEM competition, another thing I hadn't heard of. In fact, I hadn't even heard of synthetic biology. I had done research for 15 years, but that was a long time ago. So the first thing I did was call old colleagues and say, have you ever heard of this field, synthetic biology? And it turns out that at Genentech, they had just heard about it because they had a speaker the month before. Oh, yeah, we just found out about that field. So you guys are way ahead of the curve on some of the big powerhouse companies, that's for sure. And Wendell proposed putting a team together for iGEM. And I'd done enough research to know, after reading his email, that it was a pretty high-end thing he was asking us to do. We were used to sending students for summer internships at UCSF, and they had been very, very successful because we have a molecular biology program at Lincoln High School. But this, I felt, was another order of magnitude in terms of a challenge. But if they were willing to take a risk, we were willing to take a risk. So my attitude was, c'est la vie. Let's give it a shot. The worst that could happen is we fail, and we learn from it. Well, as it turns out, we did very well. We were honored to make the top 6 finalists at the iGEM competition last year. It was a great experience for all of us, especially our students. And Wendell invited us back again this year, so we have another team going to Boston this November, and we're hoping they do well. Throughout the conference, high school has come up several times, and with an understandable degree of skepticism. A lot of the questions have, well, how much are the high school students really contributing, or are they just following the instructions and being told what to do? So I thought the best way to show you that they are actually capable of competing is to show you the curriculum itself. And I think once you see that, you'll see that they do have the background to actually contribute in a major way. So at Lincoln, we have actually 2 years of molecular biology. We started with a first-year course in 1995, 1 class, and now we're up to 5 sections of that first-year course. There's 150 students a year taking this first-year course at Lincoln. And I always used to say, gee, at the end of the year, if I could only keep these kids for another year, they're so well trained after the first year, the things they could do now. And fortunately, thanks to student demand, we do now have a second year of molecular biology at Lincoln for kids who have completed the first year, and it's all project-based. And that is where the iGEM team comes from. Okay, so we have 180 students a year now taking biotech at Lincoln, which represents about half the graduating class. Okay, so what I want to do is race through the curriculum for 2 reasons: time, but also because it will be very familiar to all of you. And it'll give you some idea what their background is. Okay. Well, we start out with an introduction. What is biotech? They understand the technology part. They know bio has to do with life science. But I start out by talking about the industry, because I worked in both academic and industrial laboratory, and it's a very practical generation nowadays in the Bay Area. Practical meaning they want to know how it relates to making a living. So we start by talking about the 3 phases of biotech: research, that it's an applied science, and an industry. And we go through the industry pyramid from step-by-step research, development, preclinical testing. They understand what phase 1, phase 2, and phase 3 clinical trials are. They understand what an IND and PLA filing is. And at the top of the pyramid, of course, is the almighty dollar sign. I start out by giving them a very basic definition of biotech, the use or alteration of cells, molecules, or organisms to accomplish something useful. Well, this is really not so modern. Ancient people have been using biotech for thousands of years without knowing what was going on, really. Selective breeding is an obvious example. Making cheese, yogurt, fermenting wine and beer, all of these involve the use of microorganisms to accomplish something useful. And then we give a little introduction into the molecules, of course, mostly focusing on DNA and proteins. Now, to give it a human face and show them why it matters, we want to establish a context. We start out by studying 12 different genes. And I think the best way to find out what genes do is to show what happens when they don't. In other words, I picked 12 genes we all have, and I talk about the diseases that happen when there are mutations in those genes. And we start out with Lorenzo's Oil, which is a great movie about ALD, an awful X-linked recessive disorder. And that really gets them hooked. And then cystic fibrosis will be the main disease we focus on because a lot is known about CF. And then the other genes include the PAH gene, associated phenylalanine hydroxylase gene, associated with PKU, factor VIII gene, associated with hemophilia. I picked a bunch that are both enzymes and structural proteins. The fibrillin gene, for example, is associated with Marfan syndrome. The fibroblast growth factor receptor gene is associated with achondroplasia, a type of dwarfism. And what I'm really after here is to show that there's a human side to all of this, that this matters to people's lives, and also to illustrate what it is that genes do. And then we talk about how good genes go bad with in vivo mutagenesis. Okay. We focus on CFTR because the structure of the protein is known. It's a transmembrane conductance regulating channel. It's a chloride ion channel. So we can talk about what the protein looks like, what its function is, and how when it is, there's a mutation in that gene, how the shape of the protein changes in such a way that it can no longer function. And then how biotech impacts CF treatment. There's a really terrific little therapy that came out of Genentech. It's a DNA ACE, actually, because Steve Shack had the bright idea that a lot of the mucus associated with cystic fibrosis was actually lysed bacteria in the airways, and lysed bacteria, DNA, and it turns out that if you inhale DNA ACE, it loosens the mucus and makes it easier to expectorate. And then we construct and interpret pedigrees, and of course they're very interested in sex determination. We talk about Klinefelter syndrome and Turner syndrome, and this interesting gene on the Y chromosome, the SRY gene, that's sort of the master transactivating gene that determines maleness. Going a little deeper, we talk about factors beyond simple Mendelian inheritance, polygenic and complete dominance, multiple alleles, and of course epigenetics, so interesting right now. Amazing, really, how much the environment not only interacts with our genes during our own lifetime, but leads to heritable changes. And now, history. Oh, the big H word. Students hate that word usually. I love studying history, but it's not especially popular with young people. But the reason I wrote this part of the course, very important, there's a question that high school science teachers dread getting. Mr. C, I understand the concepts you just presented, but how do they know that? Typically, the answer to that will be, well, it took years and years of experiments that I don't have time to get into. What it really means is, I don't know. So I wanted to show them how we got where we are today by starting with experiments that they could easily wrap their mind around. Darwin, his most sophisticated tool were a pair of binoculars. Mendel, a little more sophisticated, a scissor and a brush to control the pollination of plants. And then we move on to cells and the experiments of Griffith and Avery and Hershey and Chase, et cetera, et cetera. And by the time we get to Watson and Crick's model, they finally can see the thread of how science is a bunch of observations that build on observations that came before. And finally, they can understand, now I see how we know that. I think that's a really important point. Okay. Now we get into the molecular biology part of the course. And here is our textbook, the New England Biolabs catalog. How many of you use it? I'm just curious. Okay, so that's our textbook. I have a class set of those. And we learn quite a bit about restriction endonucleases, where they come from, what organism, how they cut, what buffer is required, what are the elements of the buffer, how an 8-base cutter cuts less frequently to generate larger fragments. It turns out there's so many great tables in the back of that catalog. It makes a great textbook. And restriction mapping and electrophoresis, obviously. Then we get into replication, including the life cycle of the cell, mechanisms of replication. This is important right here. You'll notice that we spend a week on this. 7 proteins involved in vivo replication. So they learned about helicase, primase, the 3 different activities of DNA polymerase. It's exonuclease and its polymerase activity. They learn about primase, the RNA primers that are put down, and how polymerase can chew those away and fill in with DNA. Telomerase, topoisomerase. That's a lot of detail for high school. The reason, though, is that if you show them how it works in vivo, they can actually come up with the PCR technique on their own, which makes them feel very smart. We talk about, well, how would you compensate for these 7 proteins when you're only using one of them in vitro, being DNA polymerase? And just incredible amount of thinking that they can apply to it. Every single year, they think of a way to compensate. Obviously, heat easily compensates for helicase, for example. Then we talk about the errors in replication and how those lead to mutations, the different types of mutations, and then, of course, the all-important PCR technique. Now, with gene expression, we talk about the differences between DNA and RNA, structure of gene, how they're regulated, what happens at the 5' end, what happens at the 3' end, how it's simple. We use the operon model for prokaryotes because it's such a simple model to follow. And then we don't say a heck of a lot about this. They know about transcription factors, enhancers. They know about the promoter and how that interacts. And then post-transcriptional modifications, of course, would be how RNA is processed. When we talk about translation next, using the codon tables, the requirements for translation, the structure of the ribosome, transfer RNA, and mature RNA, how RNA is spliced to give different versions of a message. Then protein structure function, the relationships between. They know about the classes of the amino acids, hydrophobic, hydrophilic, charged. And we talk about the bonding, of course. They know the peptide bond, and we talk then about how you can take advantage of these qualities of proteins to do protein purification. We talk about 3 different kinds of chromatography: hydrophobic chromatography, ion exchange chromatography, size exclusion. And then we end with ethics, of course, because it comes up all year long, and this is part of what hooks students. So we actually debate a bunch of different scenarios: genetic privacy, and we end with one of the great science fiction films, I think, of all time. If you haven't seen it, you absolutely must. Gattaca, which brings up an incredible number of issues. Now, I'm going to show you a little bit of detail on 1 or 2 of these units, because this is just an overview right here, so that you get an idea of how much detail they get into. I should mention that in addition to teaching applications, what really gets students interested is implications of this technology. What does it imply for the future? What does it imply in terms of ethics? What does it imply in terms of what we might be able to do someday? And then also the limitations. The minute you talk about limitations with high school students, for example, we'll talk about the limitations of the PCR reaction. You really can't easily get fragments bigger than 5 kb, let's say. And they want to know why that is. Have they tried this? Have they tried that? That's what really gets them, I think, involved. All right, now I'm going to just go to one of the units to show you the level of detail that they learn. So here's some detail on this unit. We talked about this. Let me go a little further. Tissue and cell culture. Informatics. I haven't talked about that one yet. So they know what RFLPs are and VNTRs are. They do BLAST searches. They're familiar with EMBL Swiss database, and we use both CHIME and RASMOL to show how proteins fold and protein modeling. So there's a little bit of example of the level of detail. The last part of this presentation I'm going to show you is the labs they do, because that is really what trains students in terms of how to do stuff. So here are the traditional applications. We make cheese and we make yogurt in the laboratory. In both cases, we use bacteria for that, but we also use the enzymes to show how we've improved upon that by isolating the enzymes from these bacteria. For example, renolase from the stomachs of cow cells. We breed fruit flies. That's our way of demonstrating Mendelian inheritance, and also it hooks in nicely with Thomas Hunt Morgan, which is one of the scientists that we study on the road of the history. Actually, this is maybe the hardest thing right here. We're the only country in the world that measures things in gallons. When I first arrived here, I said, wow, gas is cheap in Hong Kong. Then I remembered that we're the only ones that use gallons. It was in liters. So I'm a victim of that myself. Microbiology, we culture bacteria, identify plasmid containing E. coli. We isolate genomic DNA from 3 different sources. Their own cheek cells, epithelial cells, from a kiwi. I used to use onion, but kiwi smells a lot better. And also from bacteria. And then we run our first gel. What does genomic DNA look like on a gel? Well, it's a big smeary streak because it's such high molecular weight DNA. And then we do our first transformation with a plasmid p-amp. That the only gene on there I care about is ampicillin resistance gene, because this is to demonstrate Griffith and Avery's experiment that proved that DNA was the transforming principle. Then we cut lambda with some different enzymes and map it, and the Phi-X174 genome, it's a really small one. Then we ligate those fragments back together, and we run our second gel showing both before and after, so the activity of ligase is demonstrated very nicely. Then we get into PCR, which we do 3 times in the first year. The first time we just amplify a fragment from lambda, because the goal here is just to show how it works, and we run our third gel with PCR reactions. They will later on do an ALU from their own cheek cell DNA. There's nothing more interesting to them than to look at their own DNA, obviously, and ALU is polymorphic at the loci we use. Then we get into our protein purification unit. We do a little we do PGLO is a funny name for a GFP-containing plasmid. So they do a transformation, and then we grow a little culture, and then we purify it partially using HIC chromatography and run our first polyacrylamide gel. This is a great little unit right here where I present them with a problem. I say, well, you know, the shampoo that we sell is burning people's hair and causing dandruff and itchy scalp. What's the problem? The problem is there's a contaminant. And they have to figure that out by running gels, and then they have to figure out a way to purify out the contaminant using both size exclusion and ion exchange chromatography. Here's the polymorphism I talked about. The one at ALO, and DS180 is a very interesting loci because there's many, many variations on that one, and there goes agarose gel 4. This illustrates an important point. A lot of my students say that in 4 years of an education at UC Berkeley or UCLA, they only do these techniques 1 time. Well, if you do it once, you sort of get an introduction to the theory, but if you do it 4 or 5 times, well, then you know the difference between TAE buffer and TBE buffer. What are the advantages of each? I should mention, I teach everything the old-fashioned way, because when I did molecular biology, we didn't have any kits. We made our own buffers, so I want them to understand what is tris, what is EDTA, what's the differences between Triton and SDS as a detergent, because I think kits get you away from the fundamental understanding of what's going on inside of these buffers. I think it leads to great thinking. So even though you could do all of this now by adding buffer A, buffer B, and buffer C without knowing what's in that, I think it's critically important to thinking to know what's in that. Then we do an ELISA. We do an ELISA on fish muscle extract. Oh, I'm sorry. We do an ELISA on, this is actually not a real, it is a real ELISA, But I couch it as though it's an infectious disease that we spread through the class. I spike one sample, and then people swap fluids, and we see the epidemiology within the class of how that spreads around using the ELISA as the test. And then bioinformatics, we sequence our mitochondrial DNA, a region in the D loop, and then we go online to the NCBI website to enter our sequence and see if we can find that indeed it is the mitochondrial DNA D loop. Okay. Now, in the second year, this is all project-based, so I'm just going to show a couple of these. We plate libraries. I don't know if anyone uses Lambda GT10 anymore. I might be dating myself here, but this is how we used to screen libraries when we cloned genes way back. We do southern blotting and western blotting. Here's where we make fish muscle extracts and run them on a gel. Myosin is highly conserved protein, so every single fish has a big fat band there, and that's what we do the western blot on. And also you can infer evolutionary relationships looking at the different fishes and the commonality in their banding patterns in terms of protein. And then, this is an interesting one. We go to the store and find corn-based products and see if we can find the BT gene using the PCR technique to show that it's pervasive in corn in the United States. You can't even get a corn-based product in the U.S. that is not genetically modified. We make recombinant DNA starting from scratch. I give them some bacteria, they streak it out, they pick colonies, they make the DNA, purify it, cut it, paste it, and ultimately transform it. And then we do a little PCR cloning. I teach them how to use primer design. Wendell has been fantastic at helping us with that because we never used yeast very much, but we go to the yeast genome database to do this. And that's a little overview of some of the labs. Okay. I think if you look at all of this, you realize that these kids are coming into Wendell's laboratory actually knowing how to do stuff. One of the reasons I designed the course this way, I heard 3 complaints from academic professors such as Wendell or the biotech industry. There are 3 major complaints they have about incoming undergraduates. Now, this is someone with a 4-year degree at a university. Number 1, they don't write very well. Well, that's no surprise. That's a pervasive problem. Students today don't read for pleasure at the voracious rate that we did as kids, so they don't write very well. So we have a lot of writing in this course. The lab notebook will be over 200 pages of writing by the time they're done at the end of the year. The second thing is they don't know how to work in teams. So I have 6 teams in my classes. Each one is a biotech company. They visit the company. They study its products. I make them understand the mechanism of action of a product. An example would be Avastin. That's a good one for me to give because I actually cloned the VEGF gene when I was at Genentech. So they see how the anti-angiogenic molecule will work. Mechanism of action, I think, is what's most interesting. And then the third thing they don't know how to do, here it is, someone who went to UC Berkeley or UCLA for 4 years, eager to get working in your laboratory, but they don't know how to do anything. Whereas Lincoln students know how to do stuff. Okay. I want to end to give some time for questions, but I have to give a huge thank you to Chris Voigt for inviting me to this meeting and Drew Endy for his wonderful comments that made it into the New York Times and the San Francisco Chronicle article about us. And Lauren, thank you so much for helping us get here. Obviously, these guys right here are what it's all about. Wendell Lem is my new hero right here. This guy was willing to take a major risk allowing high school students into his laboratory. He wasn't sure if they would just break everything or be an absolute disaster, but I'm delighted to say that the entire LIM lab tells me that they not only enthuse the laboratory, but they actually work pretty independently after a week or 2 of getting on their feet. So that's the overview of the program at Lincoln, and I'm delighted to take any questions you might have.
Thanks, George. It's really wonderful. I hope that a lot of universities would have the same kind of curriculum in their freshman years.
Thank you.
Okay, we can anything, some question? Yes?
For someone who might be interested in implementing this curriculum in a high school, what's the financial outlay for a program like this, equipment and reagents, compared to, say, more traditional high school classes?
That's a great question. We've got about $500,000 of equipment in the lab at Lincoln. So we're actually better equipped than some of the smaller academic laboratories I've seen. For example, we have 4 9700 thermal cyclers. But it's mostly used equipment from companies and universities. Their garbage is our treasure. So I'm shameless about asking for things. The great thing about working with kids, you can be shameless about asking for stuff. So I have so much extra equipment that I'm able to equip 5 other high schools now in San Francisco that have adopted the first year of our curriculum. My goal is that eventually every single high school in San Francisco offers at least the first year of the course. I should also mention I write a grant every year to the Genentech Foundation for $50,000 that helps me buy all the consumables and do professional development. Now, what if you don't have all of this accessible? Well, almost any high school has a university not too far away. They should tap into those guys, either for loaners, loan me a PCR machine, or it's just amazing how much waste there is at the university and especially in industry. So we're able to get tons of free stuff that way. But I should also mention that writing grants for this kind of stuff is really easy if you're a high school teacher, because the funding agency think it's quite cute that kids are doing stuff like this, so it's pretty successful at getting funding. Yes, Rick?
I'm wondering what the distribution of your students were about GMO.
It's a very skeptical generation about stuff like that. But on the other hand, they grew up in this day and age. So I always have a small percentage of people that are against it, just for the same reasons, say, the Europeans are skeptical about it. They understand, for example, that you can't isolate a crop field over here that's got BT corn and have organic farm, 2 miles away. There's going to be cross-pollination thanks to bees and stuff like that. By and large, they have no fear of technology because they grew up with so much technology. So I would say that mostly they don't worry about it.
Thanks. Do you find that it's really just your brightest and best students that you have on this course, or is it more inclusive? Do you manage to drag more students into it?
Well, if 180 students a year are taking this course at Lincoln, I should mention we're a meat and potatoes, comprehensive, non-elitist, take-all-comers high school. Nothing fancy about our school. So if half the graduating class is taking it, you can believe I have a bell-shaped distribution curve. I don't assume any prior learning. The only prerequisite is having taken basic biology and getting a C. Well, that's pretty reachable to a wide range of students. But I should mention that we certainly have superstars who wrap their mind around it quicker. And the other thing, here's why it's really important to have biotech in high school. We're missing the boat on a tremendous amount of talent that might go into this field because they don't know about it. So we have, I'd say, in a good year, 50 to 70% of my graduates go on to study molecular biology in high school and college, which is pretty remarkable because the national average is less than 1%. Sir?
So how about the faculty members or the teachers?
Oh, that's
ready for taking up such.
Here's what I love that question. Here's why. So I can't teach all the sections anymore. It's beyond my full-time load, so I had to train another teacher to do it. She's a French literature major. So how did she do it? She took the class for an entire year. Her name is Julie Reese, I should mention that. And she teaches now 2 sections of the first year course. She actually sat in the classroom for the entire year. It's not a practical model because it's expensive. It requires the school district to pay 1/5 of the salary so a teacher could sit, but it pays off so well. And she's doing a great job now of teaching that. I really feel that teaching is the harder skill. Science can be learned. Sometimes we get a very elite view, highfalutin view of ourselves that this is not rocket science, is the way I like to put it. It can be learned.
Any more questions?
You may have said so at the beginning, sorry I missed. How many hours does this course involve?
Okay. It meets for about 5 hours a week, so I have them an hour a day, 3 days a week, and then I have them an hour and a half to make it easier to do the long laboratories, and it lasts 1 whole year, the first year. The second year course meets a little more. It's at the end of the day, so we can run over into after school, because when you're doing things like Western blotting, you're not going to get that done in an hour and a half. So both of them are full year courses with a minimum of 5 hours a week.
You don't have never had opposition from the school direction for being too much at the expenses of other classic.
It is a problem, I would say, in that since it's such a popular elective at Lincoln, you would think, well, gee, is that going to cause a reduction in some of the other electives? But here's the funny thing. It's had the opposite effect. We have most students at Lincoln take 5 or 6 years of science in 4 years because they're interested. So it's actually increased enrollment in sciences throughout. And I should also mention that one of the reasons that this can happen is there are no standards written for biotechnology in high school. So I'll be dead probably by the time they write those, because it's going to be way in the future. And that frees us up to do what we know is good. I think standards actually eliminate what we can do. I'm fearful of the standards. I think they're poorly written, and I think they're very low expectation. What would you rather have? Someone in your laboratory who can answer multiple choice questions, or someone in your laboratory who can design a controlled experiment?
Okay, I guess we need to move on. Thanks, Joe.
Thank you.